And hello everyone. I'm Nikki Newton from the Ecosystem Analysis team at JNCC, and I want to welcome you to the final event in the 2021 TEPoP Festival, where we've teamed up with the JNCC Science Talks once again. So to round up this year's proceedings, we've got some talks lined up about developing biodiversity indicators in two different contexts. So indicators are obviously a really influential way in which biodiversity can be presented to demonstrate biodiversity's responses to pressures or conservation efforts, and also to trigger action off the back of these findings. So we're looking forward today to hearing more about the processes involved in ensuring they're developed rigorously to deliver for different scales and for different reporting needs. So I'm aware that we might have some people on the line who won't be familiar with what TEPoP is. So here's a quick summary slide. Sorry if it's familiar for everyone that's been on for multiple events. So the organisations in the box on the left in various combinations fund, organise, run and steer the terrestrial monitoring and surveillance in the box on the right on this slide, all of which engages volunteers to undertake the majority of species recording involved in the various schemes. So many of the challenges or opportunities relevant for engaging volunteers with monitoring one taxon are also relevant for other taxa. So it's beneficial for us to work together, to collaborate, to exchange knowledge and to sort of problem solve together. So we aim to do this as the UK Terrestrial Evidence Partnership of Partnerships, or TEPoP. So as I said, this is the last of the TEPoP events in the festival this year, and we want to thank everyone who's participated in any of the events. And we've had some really excellent discussions and heard some very interesting presentations. Recordings of all of the events so far can be found on the JNCC YouTube channel, and a recording of this event will also be added there. So if you want to find out any more details about anything that we've discussed in the events, or give any feedback or join the TEPoP bulletin mailing list, then please do contact my team at the TEPoP mailbox, and the address is on the slide. And as I mentioned, the TEPoP Festival has teamed up with the JNCC Science Talks this year for some of the events. And the Science Talks run fortnightly on Tuesday lunchtimes, and many of the events are open to audiences beyond JNCC staff. So if you're not a JNCC staff member on the line and you're interested in being added to the mailing list for the JNCC Science Talks, then please do email the address on the slide for the Science Talks. And coincidentally, the next Science Talk on the 16th of November is actually also on more work that JNCC has done developing another indicator, one on global environmental impacts of UK consumption. So if this is of interest, then do please get in touch for more details on how to join. But now we'll move on to our first of today's talks. So this is from Dr. Oli Pescott, who is a data analyst and plant ecologist at the UK Centre of Ecology and Hydrology. And Oli's going to talk about developing the new UK biodiversity indicator, Plants of the Wider Countryside, which uses data from the National Plant Monitoring Scheme. So I think this is a really exciting topic because I was involved with the launch of the NPMS as part of my first job when I started at JNCC. And now to get to the point of having enough data from the scheme to have developed a new indicator from it 6 years down the line feels like a really great success. So over to you, Oli, to hear more about this. Thanks very much.
Okay, thank you, Nikki. Can you see the main screen rather than the supporting one, as it were?
Yes, we can.
Brilliant. Okay. Well, yep, thank you for that introduction. I'm going to give a broad overview of the NPMS very briefly first. I imagine a lot of you will have heard me or others talk about the NPMS, so I don't go into great amounts of detail about the fundamentals of the scheme. I just have a couple of slides on that, but there'll obviously be time for questions afterwards, so hopefully I won't gloss over too much important detail. But really, I'm going to try and focus on the development of the indicator, as Nikki said, which was first released as an experimental statistic last year, and then go on to talk about some broader considerations for addressing sampling biases, which are important for the next phase of development of the NPMS indicator as we try and move on from it being an experimental stat to it being a full biodiversity indicator as part of the JNCC DEFRA set of UK biodiversity indicators. So some broader context first. Obviously, UK plant habitat monitoring has traditionally focused on species distributions at very broad scales. The classic example is illustrated here on the right-hand side of this slide, being the Atlas, the British Flora, published by the BSBI and BRC back in 1962. But even at that time, there were aspirations as part of that project to actually record habitat information in the form of habitat associations. So the original field cards that were sent out for surveyors during this project, the guidance that was sent out did contain guidance about recording habitat. Some interesting correspondence here, just a bit of a historical aside between Hef Warburg at Oxford and Max Waters in Cambridge relating to how that we could elaborate perhaps on the habitat information. And that information does exist on the original field cards for the atlas, which have never been digitized and which just sit in a store here. So that information was never really used upon, even in terms of developing plant habitat associations. So there was always an interest in habitats and how plants formed habitats, formed communities and all of that, but there was never the resources and interest in really taking that forwards. So it took quite a long time for any real kind of habitat focus to be developed in the context of UK plant habitat monitoring. And after the new atlas, the repeat atlas of that first national atlasing effort back in 1962, the new atlas then in 2002, and the BSBI local change project, which resurveyed a set of a random stratified or systematic random stratified set of 2 kilometres squares across Britain between 1987 and early 2000s. After those two kind of distribution focused projects, it was increasingly recognized that actually it'd be really nice also to have information on habitats at small scales in terms of the distribution of communities and habitats, but also in terms of species compositional change. And there was actually a review of this topic in the final chapters of the Local Change book published in 2006, where they included this nice little table looking across distribution focused projects such as the New Atlas and the Countryside Survey, but also smaller, more focused surveys such as Common Standards Monitoring, County Rare Plant Monitoring, Registers, Local Change itself, and then the Countryside Survey and the Common Plant Survey, which Plant Life were running at that time. And they looked at all of the attributes of those schemes in terms of distribution change, fine scale abundance information, change in habitats and all of that. And it became quite clear that there were some quite big gaps in terms of, particularly in terms of abundance monitoring, but also linking that abundance monitoring to actual habitat change and turnover in specific habitats. Of course, the Countryside Survey existed, but that was a random stratified sample that was tended to miss some of the rarer, more semi-natural habitats of conservation interest. And of course, at that time, it was only repeated every 7 to 10 years. So this need for a more regularly updated information about plant abundances in habitats was flagged up in the UK Terrestrial Biodiversity Surveillance Strategy in 2009, like a kind of strategy document that JNCC published after lots of consultation work. And then Kevin Walker at the BSBI and other people published a JNCC technical report in 2010, exploring lots of design options, leading to some field trials, and then a soft launch of the new National Plant Monitoring Scheme in 2014, which was built on top of the what was then the Plant Life Wildflower Counts Survey, which is itself an evolution of the Common Plant Survey. So there's a kind of hybrid year where we tried to bring people across onto the new, hopefully more rigorous and more statistically robust National Plant Monitoring Scheme. So as I say, the main focus of this new scheme was to be habitat-based, to look at plant abundances within those habitats, to use sets of indicator species that were hopefully indicative of good condition of those habitats. And of course, there was a lot of careful design so that we, as far as possible, were working on a robust, structured basis to minimize bias. We also had lots of participation considerations, which I'm not going to go into account, but people can take part at different participation levels depending on their species ID skills, their habitat ID skills. So there's also so there's a kind of hybrid approach of co-design, trying to get all the statistical rigor, but also allowing for as broad participation as possible, which is also important for bringing people across from the wildflower counts and the Common Plant Survey before that. So just very briefly, as I said, focusing in on a couple of the key design features for ensuring that it was hopefully as robust as possible. There are two kind of survey levels. This is quite common with JNCC supported structured monitoring schemes aimed at volunteers, one of which relates to those 1 kilometre grid squares which people are sent to in the first place. And the three maps of the British Isles on the left there kind of illustrate what we did on that front. So the map on the far left, which is like a heat map, that was the probability surface that we developed for our weighted random square selection. So the idea here was that we wanted to focus on the semi-natural parts of the countryside rather than just having a totally random selection. And so we developed this probability surface which upweights squares which in theory have larger areas of nationally rarer habitats using land cover information. So in theory, you're more likely to be allocated a square that has a larger area of a habitat of more conservation importance. Then we use that probability surface to randomly select a tranche of 2,000 squares. In the first instance, that's the map in the middle, that central map with the black dots, and that was stratified by 100 kilometre squares of the national grid, national grids, I should say, to get even geographic coverage of Britain and Northern Ireland. And then on the right-hand side there, the final map is the allocated squares in the first 2 years of the scheme, the red dots. So obviously there's some uptake bias, geographic bias in survey effort, which I'll come back to later on. The right hand is obviously zoomed in on a 1 kilometre grid square, and here we're looking at the selection of plots within the 1 kilometre square and the efforts we went to try and make sure that was as unbiased as possible. So each volunteer receives this type of map of their square. The numbered little square plots are locations where we suggest people locate square habitat plots, which are focused on extensive habitats. So again, that's to minimize bias in plot placement. The linear plots are intersections with the grid lines where the red stars are, and they're again to minimize bias in plot selection in linear habitats such as streams, arable field margins, and hedgerows. So that was an effort that was gone into to try and develop a robust basis for the scheme, and we'll come back to that later. But as obviously we're collecting then all this information on individual species abundances in small plots within particular habitats. So somebody goes to woodland, they set up a 10 by 10 meter quadrat. Depending on their level of participation, they're looking for either some subsets of indicator species, or if they're an expert, they're looking for all species. And we ask them to do that twice a year, which is what's this part of what this cartoon illustrates. But of course, in developing an indicator, we also need to take into account certain biases which arise in the data collection process. The one which people talk about most frequently is detectability. People go to a survey location, they do something, but inevitably they miss the presence of some species. They misestimate some abundance. So the information you get back is not truth. It's been through some sort of filtering process based on some interaction between the environment and the surveyor. So surveyor expertise, whether a species is above ground at that time of year, whether it's died back and is no longer identifiable. So the fine detail of this cartoon here, which I won't go into, is just to illustrate that point that there's a difference between the naive stats that we get back, the naive information we get on plot occupancy, that we get on annual cover of plants in plots, and the discrepancy between that and the truth, the true underlying occupancy, the true underlying percentage cover, which we would have if we could extract perfect information on the environment by whatever means. So our indicator tries to adjust for that detectability process which happens in the field. And again, I won't go into great detail on this, but the plot link, the link to the title of the associated report is given at the bottom there. But we're essentially constructing this statistical model which has two parts. Each part is part one is to the left of that central Z, which is the true species presence absent in a plot, and then a part on the right. The part on the left is your classic occupancy model, where you've got some variables and other things that influence as a species detectability. That influence is obviously whether it's detected on a visit. Remember, 2 visits to a plot for a year. And from that, we can infer something about the true presence absence condition of that species in that plot in that year. But then on the right hand side, we also have the percentage cover data, which people record according to an interval censored scale. So rather than estimating 32%, they use the category which covers 25 to 33%, whatever it is, and write a number on a form. and that's just a simplified recording process. And from that information for a species within a year, we estimate a percentage cover distribution, and then we combine that with the presence absence information, and we end up with for that species in that year within that habitat, we end up with something like a zero inflated percentage cover distribution, and the mean of that is our indicator for that species in that year. So you can imagine over time, say wild garlic in woodland habitats has a percentage cover taking into account the zeros as well in this year of 5%, the next year it's 10%. And this histogram here just illustrates that concept for any given species in any given year. And the red point there is the mean, which then goes forward into our habitat indicator. So obviously we do that across all positive indicator species in a broad habitat type, and then averaging over those species trends over time, we create the broad habitat indicator, which, you know, is very much in line with what's done in lots of other schemes, structure monitoring schemes that JNCC supports. So the four that we focused on for our experimental statistic last year were the four most widely surveyed broad habitats. So this is done at the broad scale to maximize the information content of the indicators here, rather than breaking it down into the constituent habitats. For instance, broadleaf woodland and hedges can be broken down into wet woodland, dry deciduous woodland, hedges, but here we've done the indicator at the broad level to maximize the amount of information and minimize the number of indicators for interpretation as well. The maps there show the locations of the plots that were included, and this is data from 2015 to 2019. And again, you know, you'll be able to spot, if you know distributions of habitats, that obviously there are slight biases in the data that have gone into these indicators there. The numbers on the plots are the number of taxa that have contributed to any given trend over time. So this is where we currently are. This is what's published on the JNCC DEFRA website pages about the UK Biodiversity Indicator C7 plant to the wider countryside. And in the technical documentation supporting this, we discuss what will be needed to bring this statistic to a full official statistic within that indicator set. And really one of the main things that needs addressing is the fact that structured schemes still obviously have sampling biases. Yes, we've put that robust basis in place as far as we can. We've got a clear probability basis to the selection of 1 kilometre squares. We know how the strata work. But even so, obviously, and we're accounting for those, trying to account for detectability issues at the small scale. But even so, people are not surveying Britain and Northern Ireland evenly, of course. There are uptake biases based on where people live and work, which places are easy to access, and all of those types of things. And we investigated that in our first paper on the design and assessment of the scheme, how it's performing in the first couple of years. And yeah, that was related to population density, as you'd expect here, both the proportion of squares allocated within a 100 kilometre grid square and the proportion surveyed. There was some relationship with population density. Obviously, there'll be other factors as well. So we really need to take this into account in order to have a truly representative national indicator. Because at the end of the day, we are presumably producing UK biodiversity indicators to be inferential statistics, not merely descriptive statistics. And the difference is that if we were merely saying that our indicator was only indicative of changes in those surveyed locations, then it would be a descriptive statistic, and it wouldn't be something that we could base inferences about the whole of the UK on, because that would require extrapolating outside of the sample. And inferential statistics are all about making broader inferences based on samples. And statistics for broad inference need both internal and external validity. And these are concepts that are quite well known in medicine, in the social sciences, but they've not received as much attention as they should have done in ecology and conservation. The idea is that internal validity is about how some individual, say, sampling program or experiment is set up. Say you might do a randomized controlled trial of some conservation management intervention, and if you've done the randomization properly and all the other things, there's controls, then those data are fully appropriate for estimating a causal effect of that management treatment. And that's internal validity. The data that you've got in hand can tell you something about that sample. But if we're interested in inferential statistics, we're also interested in external validity. Are those estimands that we've calculated actually representative of some wider population? For instance, we've done some experiment here, but is that actually telling us something about the wider population of units that we're interested in? Is this heathland site representative of all heathlands? And obviously this is something that people think about a lot in terms of electoral sampling. So if you're asking people how they vote and you get some information back about it, is that really externally valid? Can you make a good prediction about electoral outcomes based on that? Often, predictions are pretty poor because the external validity is not there. And the only way to get true external validity is to have a full probability sample of your population of interest, or at least the best way, the most efficient way. And that's, of course, often very difficult to do. And as we've just seen, we almost never achieve that in biodiversity studies, particularly with these type of volunteer-based biodiversity indicators. So really any indicator that's claiming or is implied by saying it's a UK biodiversity indicator or whatever, if it's implied to be inferential, it should have some sort of transparent and clear assessment of its proposed or purported external validity attached to it. And myself and colleagues have been thinking about this. One last point about potential biases. So you can adjust for one potential bias, but it's unlikely to sort out all the issues that you might have. For instance, the NPMS indicator, as I've said, has this measurement error model where we're trying to adjust for detectability, sometimes called an occupancy model, and we've integrated that into a larger framework. And of course, that might work very well for any given species data set. There's that internal validity. So you might be very confident that you had adjusted for detectability in that set of plots that you had data for, and therefore you might be very confident that your true occupancy estimates for that set of plots was correct. But there's absolutely no reason to suppose that that adjusts for the fact that you've sampled the country unevenly, which is the external validity component. So different potential biases require different modelling fixes, adjustments, and obviously checks on whether those have actually worked. Of course, the alternative is just to acknowledge that there are limits to what you're saying, and obviously that's quite important as well in many cases in terms of titles of papers, abstracts, communicating conclusions in academic work, and all that type of stuff. And so I was thinking about this with some colleagues here and elsewhere outside of CEH, and we started to look at other disciplines and think about what happened outside of ecology and conservation. And there are many good models for this outside of ecology and conservation, where other disciplines have clearly considered how to transparently report on sampling biases, particularly those which affect external validity, but also the internal stuff about risks of bias in randomized controlled trials, in other observational study types. And across medicine, epidemiology, public health, psychology, and the world of systematic reviewing, so evidence synthesis, there are quite a lot of tools out there which give structured frameworks, performers quite often, for assessing the risk of bias in some conclusion that's been reached from some set of data connected to some model and some sort of inferential goal. And I won't go through all of these here, but they're listed. Some of them you may have heard of, perhaps they're all new to you. A lot of them have the abbreviation ROB for risk of bias. So like Robin's eye at the bottom there is about risk of bias on observational studies of medical interventions, for instance, where you've just got some observational data that wasn't experimental and split it up into exposure to, I don't know, indoor smoke and non-exposure, and so there's no kind of good randomized controlled basis, and you're just trying to make causal estimates from those observational data. And so most of these tools are actually kind of structured question-based things, so they're qualitative on the whole rather than quantitative, or they can have quantitative support elements where you might need to calculate something to estimate the risk of bias, but generally they're about encouraging thoughtful engagement with the data, thinking clearly about the inferential aims of your data and what you're trying to achieve, and then honestly assessing the risk of bias associated to various aspects of the desire to have that inferential statistic. And of course, there's a very clear link here to the Government Code of Practice on statistics as well. I've just picked out a few points here where I think these considerations link in particularly well to what we're asked to consider under the Code of Practice. Obviously, there are several pillars to that, but I focused here on quality and value. So within quality, there are things like making sure that you have suitable data sources, sound methods, and assured quality. So of course, under suitable data sources, if we're thinking about inferential statistics that are true indicators nationally, then we need to make sure that the data are actually representative of the claimed population of interest. If we're talking about sound methods, we need to really be checking that there's strong evidence that a method does adjust for biases in a given data set in hand? Is the bias modeling that's been undergone clearly explained? Are you aware what the likely accuracy of the result of that process is? There are full assessment of uncertainty in all its forms. And likewise for the value pillar, for clarity and insight, if the indicator is actually unrepresentative of the purported domain, is that clearly stated? Is that, if it is unrepresentative, is that still meaningful and authoritative as a statistic? Or can it only be meaningful and authoritative if that's clearly explained and highlighted? Likewise, innovation and improvement, we're always trying to get better. So, transparent assessments of bias, actual modeling of all the biases could come under that pillar there, under value as well. So there's a lot of things where I think this links into government codes of practice. So what we've done is created the first, what I think is the first formal risk of bias tool for ecology, which we've called the Risk of Bias in Temporal Trends, or ROBIT. It's led by Rob Boyd, my colleague. So it's very nice that he gets his name in there as well in that acronym. And it's basically, again, a structured set of questions where it's designed to elicit information on this potential for bias in any given study. So there are several elements which I won't spend too much time on. We asked the researcher for a clear statement of the research aims, those inferential aims, the things that they're actually trying to say something true about outside of their sample. There's a pre-bias assessment part where people look for various errors in the data set, just checking that all the data makes sense in terms of spatiotemporal stuff, taxonomic things, all those types of things that can go wrong in processing data, making sure they understand all of those elements. And then the bias assessment itself, it's important to specify the scale at which you're making inferences, because that might certainly links into the research statement. For instance, if you want to look at change in decadal periods at the 10-kilometer scale, that has different implications for potential biases. Then looking at things at an annual scale, in say a finer scale like 1 kilometer squares. So it's very important to link that bias assessment to the research statement in terms of the scale at which your inferences are desired. And then you move on to formally assess the risk of bias across relevant domains. And those are things like geographic coverage, environmental zone coverage, taxonomic coverage, and how each of those things varies across the temporal periods that you're interested in. For instance, if you sample something in Cornwall in 1970 and then sample in Lincolnshire in 1995, unless the trends in that taxonomic group or species are completely uniform across space, which of course you can't prove anyway, there you're confounding space and time. So there you've got the risk of bias across perhaps the taxonomic domain, the geographic domain, and the environmental domain because of that temporal shift. And then the final part is saying about how you aim to mitigate or acknowledge, because you might not be able to mitigate in some cases. So they're clear statements about mitigating biases or limiting the inferential claims, not saying something's representative of the UK when you've only got data for Sussex or whatever it might be. So it's about being clear and transparent with those risks and what they mean for your inferences. And as I've kind of covered there, and this links back onto what we're trying to do for the NPMS as well, there are different mitigation strategies. So we might work at a different scale. We might find that the data are very unrepresentative at 1 kilometer, but there are more data at 10 kilometers. So we're going to make our inferences about change at 10 kilometer, not at that fine scale. You could weight or thin the data to obtain more even coverage across space, time, environment, whatever it might be. Your weighting and thinning are techniques which are very well developed in the type of electoral survey sampling that I mentioned earlier. You can have more complicated models of biases. If you think your process causing bias can be described by covariates, then that naturally fits into some sort of regression framework, whether it's a hierarchical model or a simpler regression-based adjustment. And so that might be possible as well with appropriate checks of fit for the internal validity side of things. And of course, the final one, which I mentioned, was just don't claim that your stats are representative of particular domains if they're clearly not shown to be not representative by the risk of bias assessment. Sorry, just go back. And probably for the NPMS, I think mainly we're going to look at modeling the biases by using covariates or spatial fields or something of that nature. And there's a lot of work been done in that recently in various areas. But if we can identify specific covariates, say like accessibility of countryside and things like that, then we may start to build in those covariates into specific places in the model as well. And I'm just starting that process, so I don't have anything to report on that, although a lot of the thinking has been done. And this is part of that thought process. So finishing up, just to recap what I've been saying. So the NPMS has been designed to monitor habitats with minimal bias, but sampling bias is still almost inevitable with volunteer-based scheme. It's probably true of all of our volunteer-based schemes, especially when we're considering even coverage of space across time. So the idea that every year or 5-year period or whatever it might be is actually representative of the statistical population that's implied, normally the UK or some habitat subset of the UK or whatever. And I think few current indicators really address that, if any, in a clear and transparent way, despite the kind of encouragement from the Code of Practice and other examples in other disciplines such as epidemiology, political science, etc. And so many approaches to the problem do exist, and we should take heed of them and think about how we can apply them in ecology and conservation, because ultimately we really need to catch up if we want accurate inferences with transparent uncertainty assessments across time and space, and not just descriptive statistics which are kind of slightly implied to be indicative of the UK in a slightly merely mouthed way, but which are not actually externally valid in any way that people in, say, the medical sciences or the social sciences would actually recognise. So I'll just finish with acknowledgements. I've just got all the people from the NPM, well, not all the people, because many people have contributed to the NPMS, but maybe the top recent people and the group of people who are working with me on the risk of bias in temporal trends project, which hopefully should be coming to a journal near you very soon. So thank you very much for listening.
Thanks very much, Oli. That was great. Great sort of whistle-stop tour over the past 10 years of development, which was really interesting to hear. I think next we are going to move on to James's talk. Without further ado, we'll have lots of time for questions. Well, some time for questions at the end. So please do put your questions on the Jamboard, as Josh said at the start, and I think we'll repost the link in the chat. So yes, we're now going to move on to hearing from James Williams, who is Biodiversity Indicators Manager from JNCC, who's going to describe the process of developing the new global biodiversity framework under the CBD and associated indicators. So over to you, James.
Thank you very much indeed, Nikki. Can I just check that you can see my full screen just before I launch in?
Yes, we can.
Excellent, that's all good. Right, good afternoon, everybody. Thank you very much. Nice to have the opportunity to talk to you all. I'm going to start by just going back to pick up on, it actually links very nicely with what Oli was just saying. We've got a variety of official statistics that JNCC produces, things like individual trends of birds, bats and butterflies, but also the UK biodiversity indicators, which takes quite a lot of that data and pulls it together to give you composite trends for a variety of taxonomic groups, priority species, volunteering, a whole load of things. And much of the information about the trends on biodiversity, particularly at a species level, comes from people involved in this call. I recognised quite a lot of names when I had a quick look through the list of participants for today's webinar. So good to see you all. Now, the CBD, Conventional Biological Diversity, has got 3 goals. And it's important to think about all 3 of them in terms of conservation, sustainable use and sharing of benefits as being equally important. And we've just to sort of think a little bit about history, we've had the 2010 target, we've had the HE targets from 2011 to 2020, and now we've got a new framework being developed as a post-2020 set of targets, and that of course is within the 2050 vision of the CBD overall. And the next step really is about agreeing that set of targets for the current decade, and obviously the negotiations have been delayed by COVID, so we're behind the curve in terms of where we intended to be on having these targets negotiated, but it's a really important set of processes that are going on at the moment. And there's been quite a lot of detail being developed so far, a number of meetings that have happened over the last couple of years, including in this last September, a virtual meeting, which was trying to look at how to pull everything together. There's been lots and lots and lots of consultations. Everybody has got their views. We've gone through quite a number of iterations of what the targets might look like, whether you have some sort of overarching apex target like the 1.5% that's being discussed in Glasgow at the moment, not percent, degrees, but it's actually quite difficult to summate biodiversity to just one number. The first draft of the new targets became available for the third meeting of the Open-Ended Working Group, and there's a link there on the slides. 4 goals and 21 targets rather than 5 goals and 20 targets, which is what we had for the strategic plan for 2011 to 2020. But there are some big differences, and that's coming through in terms of the difference of focus and the learning from doing that's happened over the last decade. In parallel, there's also been a lot of work about thinking about a monitoring framework, about indicators and reporting against those targets, because we need to be able to iterate that so that we can measure the targets from the get-go, rather than going, oh, yes, we've got these new targets, and now we need to work out how on earth we're going to measure them. And those things are being iterated as we speak. So just thinking a little bit about how we use evidence to measure against those targets. If we think about reporting against international targets, things like the Sustainable Development Goals, the HE targets, the OSPAR targets, we've got a set of official statistics like the UK Biodiversity Indicators, but the 25-year plan outcome indicator framework is another set. And there's some overlap between the official statistics and the reporting, but it's not necessarily completely congruent. And part of the reason for that is that governments want to be able to bring in other information beyond the indicators in terms of making that overall assessment of progress. And perhaps it's worth thinking about this, therefore, in terms of a sort of overall universe of sort of country statistics, a wide variety of things, some of which are official statistics and some of which aren't, and some of which will be used towards reporting internationally and some won't. So thinking forwards in terms of what's going on with this monitoring framework that I mentioned, in the zero draft, there was a split between headline indicators, component indicators, and complementary indicators. And the idea here is that the headline indicators would be being used to get all parties to report, and that would help to form pieces of a jigsaw that would then give the global whole. The difficulty, of course, is actually what can you produce as headline indicators at a global level. And that's the reason for having the component and complementary indicators. That's about sort of what information is available at a more limited geographical scale or on a more focused set of particular interest. And of course, the UK biodiversity indicators fit into that in terms of giving us a sort of national view at a more detailed level, perhaps for some of the species data than we're able to get at either a European or a global scale. So how many headlines is enough, or how many headlines can we do? The zero draft suggested 48. The first draft suggested 38. But many of those have got no definition, or they are still to be developed. And there is a real issue here about capacity, particularly for countries that don't necessarily have as well developed a infrastructure of biodiversity recording as we do in the United Kingdom. So I think there is a real question here about how many can all parties implement, and also an issue about what actually needs to be done at a national level and what needs to be done at a global level. So some of the headline indicators are actually things like the Red List Index, which are produced at a global level. Now, it's not really necessary to break that down to a national level, report it, and then re-aggregate it. You're liable to get a different result apart from anything else. So global indicators that are produced at a global level for global use is one aspect. Global indicators which are produced by aggregating national data is another, and that's particularly perhaps where the headline indicator concept comes in. But there's also likely to be a need for national indicators for national use, which don't actually need to be reported at a global level, because they're actually down to how are we going to make sure that we're managing our biodiversity. And if everybody gives all of the information that they've got on absolutely everything to the CBD Secretariat, I suggest that they're going to be absolutely swamped. And just to put that into context, the sixth national report for the UK to the CBD was 314 pages long. I suggest we probably overdid that, actually. Maybe we got a bit too overexcited in terms of the amount of information we tried to provide to the Secretariat. So I mentioned that there would be a whole set of meetings still going on. I forgot to mention that the next stage in the process is a set of face-to-face meetings in Geneva in January, and then that leads into the second part of the Conference of Parties, late April, early May, which is hopefully face-to-face in Kunming. And it needs to be face-to-face in order to be able to get those negotiations to actually work. Currently, the discussions have been very, very fruitful. They've been very, very rich, but we've got an awful lot of square brackets still to resolve. Thinking about this issue of the number of indicators that might be relevant, UK has put forward a sort of clustering proposal suggesting that some of the headlines could be put under the individual goals, and thereby create a set of maybe 12, 16 headline indicators, which might be a more manageable number for parties to be able to report against. Number of these are still under development, and therefore there are some questions about what it is we would be signing up to. But the idea of having some sort of clustering might help. The other side of that, of course, is a challenge of not necessarily having an indicator per goal and target. So there's quite a lot of further discussion to be had about whether this will actually work or not. But it's certainly an approach that might help parties concentrate on what are the key messages. I just want to illustrate a little bit using one of the targets, target 3, which is the 30 by 30 target some of you may be aware of already, in terms of some of the discussions that have been going on, and in terms of the way in which the negotiations still have quite a long way to run. So the 30 by 30 target at one level is relatively straightforward. You can see aspects of that relate to HE target 11. It's quite similar in a lot of ways. In the negotiations in the OEWG, we had proposals by 29 parties, 1 non-party, and 6 NGOs and IGOs, non-governmental organisations and intergovernmental organisations. Now, we weren't trying to negotiate the text, because we weren't going to be able to do that in a virtual sense, because you need to be able to say, well, I think you need to put this in and that out, and so on and so forth. And we just didn't have the time to be able to do that. So the Secretariat have pulled together all of the suggestions that have been made into one sort of composite text, which is a bit of a nightmare. And I've deliberately titled it Frankentext, because as the delegation, we were seeing this across all of the targets, and we were going, my God, this is going to be a nightmare. Now, there's a lot of duplication in here, and there's a lot of convergence, but just slightly different wordings. And in fact, it ought to be possible when we get together in Geneva in January to be able to strike out a lot of those square brackets fairly quickly and easily. I hope so anyway, because we've got an awful lot of work to do. But the problem that gives us is actually what indicators do you use to measure progress, either against that or against a revised version that may come out of this composite text. Currently, the headline indicator is about coverage of protected areas and other effective area-based conservation measures, and that's based upon the World Database on Protected Areas and Protected Planet work, and there's a lot of good data there. The OECM data is under compilation. That's one that needs more work to be done there. And that 30 by 30, I should say, is very unlikely to be met by protected areas only. It's very likely that that will need those other effective area-based conservation measures to be taken into account in order to be able to get 30% of land and 30% of sea. At a national level, we've got the existing C1 indicator, and it's quite likely that we will need to revise how we present that, perhaps to a percentage coverage. And JNCC is also working on potentially a new indicator of management effectiveness based upon the OSPAR marine protected area assessments. And that's something that's currently going through a proof of concept work at the moment. So in the UK biodiversity indicators, we've just published them again, and we revised quickly one of them last week to publish a new global impact indicator, which Nikki mentioned in her introductory remarks. That's a really interesting piece of work, and I thoroughly recommend you join the science talk that will be being given about that next time round. Nearly 100 organisations contribute data. There's a lot that goes into this, and as I said, many of you are involved in doing that, and we publish an awful lot of information. If you printed all of this lot out, it's probably of the order of 250 pages. So it's a fairly major undertaking to do on an approximately annual basis. And Oli was talking about the new plant indicator, and one of the things that we need to do when we're publishing indicators and when we come up with experimental statistics and then hopefully take them forwards into being new official statistics is there's a lot of idealism. We really want to be able to measure the right things and measure it in the right way. But there's also a lot of pragmatism that needs to go into this in terms of having something which is an indicator is better than having no indicator to some degree. I mean, there are some things which would be so awful a proxy you would say, no, that's not worth it. But we have, in developing the indicators, had to use a lot of pragmatism in the work that we've taken forwards. So we've got a whole lot of indicators, and we're going to need to think very hard about what it is that we do to take those forwards. So Biodiversity Indicators Partnership at a global level has, from a lot of experience of working with many countries, and this is based upon probably about 70 different workshops, come up with a sort of flow chart of how do you think about it. And it's very easy to dive into the detail of what data have we got and how are we going to present it. There's actually a really, really important question that needs to come first, which is what it is we're trying to indicate. And that links back to that iteration between what's the targets and how do you measure them. And actually, if the wording of the targets changes, that may need to change the indicators that we use to measure progress against them. So we need to do some work to publish, to review the indicators after having published them this year. Obviously, we start from where we are. We need to consider a whole lot of issues around links with the country indicator sets. We need to think about the marine indicators that have been developed over the last decade or so. We need to think about those headline indicators and what it is that we can pick up from those. So need to start thinking about gaps and whether or not we can come up with a new set. I think it's quite likely we will want to do some sort of challenge workshop when we got ideas further developed. And this is likely to be after the COP incoming. It's unlikely that we will get that done before the Conference of Parties happens, because we won't have a final set of targets until after the conclusion of COP15 in China. We're currently consulting, as part of the publication of the indicators, we put a note up to say, what impact would it have on you guys if we didn't do a publication next year, or if we only did a partial publication next year? And that's a genuine question. It's a lot of resource to produce all of those indicators each year. It's a fairly small team that we've got to do it, and we do think that there's quite a lot of work in doing this review. So we're a little concerned about trying to do both things at the same time, but the aim will be to publish a new set of indicators which may well have familiar data within it, but presented in a slightly different way in 2023. I just want to finish off with a few pictures to remind us it's not just about these graphs, it's actually about nature overall. So it's about things like the big and the small. Both the Desmoulins snail there and the golden eagle have got equal legal protection. It's about what happens at day and night, sort of butterflies and bats. It's about the marine and the freshwater and the terrestrial. It's about climate change, all the stuff that's going on in Glasgow at the moment. The purple saxifrage up on the top of the mountains, what's the status of that and how's that going to change? That's one of the things NPMS may be able to help us with in some degree. And it's about the UK's position in the world. The swallows fledging from my garage are a migratory species, and of course we've got a situation for the UK where we are both at the southern end of some migrations and at the northern end of others. So the UK does have global level responsibilities for biodiversity. It's not just our biodiversity, it's their biodiversity as well. So I'll stop there. Thank you very much indeed for your attention. I hope that's been of interest, and I'll stop sharing so I can then go back and see the Jamboard and see if there are any questions. Thank you very much. Back to you, Nikki.
Thanks very much, James. That was really interesting insight into the process there, and to hear what's coming in the next year or two. We have got quite a lot of questions. I think I might start with a general one to both of you, although this popped up during James's talk, so I might start with James. How do we ensure indicators and monitoring add more value and are not simply reporting on progress, but that they can also inform the right and most valuable interventions to improve nature?
Ooh, it's quite an essay in that.
Yeah, they're all quite long questions here.
So I think there's a couple of aspects here. Indicators are only indications. They aren't the whole picture. And that goes back to my slide with the Venn diagram in terms of other action and process that government would want to be able to report on in terms of saying we are making progress against the targets. So if you take the invasive alien species target 9, HE target 9, we've got an indicator there, B6. It shows that we're getting more impact over a longer period from invasive aliens. But there's actually an awful lot of really good stuff that's also been done in terms of thinking about pathways, in terms of the GB non-native species strategy, in terms of rapid eradication of Asian hornet, as an example, that's come in. So there's an aspect here of indicators are part of the picture, but not necessarily the whole of it. Oli, do you want to add anything to that?
I think, yeah, I broadly agree with that. I mean, they're just a tool for thinking. They can stimulate new thoughts and new ways of doing things. I mean, the failure to update or produce an indicator might itself be valuable in some cases for stimulating funding and stimulating attention. So, yeah, I mean, it was a very broad question. Yeah, they're not magic bullets. They're there to kind of help us think about things and stimulate thought. They're not. Yeah, they're not reality, but they are, yeah, just one tool of many, I think. And if they can help us think about things, whether in terms of improving the way we do stuff or in terms of actually learning about the environment, then they're at least doing some of their job.
Great, thank you. We'll go on to one targeted to Oli here. If surveyors target more semi-natural priority habitats, then these may be in better condition, so they're less likely to set up plots on poorer quality habitats. Is there a risk that resurveyors more likely to find that quality declines because you're less likely to have established plots on area which gain species or quality?
Yeah, that was taken into account during the design. So there are two aspects to that. The first one was that the scheme was specifically designed to inform us about semi-natural habitats, and that's why we introduced that probability surface for the 1 kilometre square selection. The beautiful thing about the probability surface is that it doesn't exclude lower parts of the countryside. It just makes them slightly less likely to be surveyed. So we still get that information. We know the indicators indicating those parts of the countryside. If we wanted to adjust for the weights in the analysis, we could. And then logically the resulting indicator would be different. It would be about the wider countryside. In other words, we'd have kind of adjusted for the weights. So at the broad scale, that's what happens, and that's what the scheme was set up to do. At the fine scale, the reason we came up with that grid, both the grid for the square extensive plots and extensive habitats and the grid lines for the linear habitats, that the point that the questioner made is exactly that. We don't let, in the first instance at least, we don't let people self-select plots. We try and get them to target them on those grid habitats, and that's why we introduced that probability surface for the